Hello, this is Laura Roselli with a lecture on cancer, part 4 for RCBC students, but we hope others will join us. We're picking up in this lecture with a continued discussion on tumor suppressor genes and proteins. Last time we talked about P53, and this time I want to talk to you about the BRCA1 gene. You probably have heard about this. There's a lot of talk in the news about it, particularly from patent perspectives. But BRCA1 is the name of a gene that is linked to breast cancer. Often we do name genes after what happens when they're mutated, as in this case. Everyone has 2 copies of the BRCA1 gene. Its function is to suppress tumor growth, as the name of the class of genes would indicate. The key here is that 65% of people with breast cancer have a broken BRCA1 gene, have a mutation in the BRCA1 gene. And women who inherit these variants are more susceptible to environmental carcinogens. So remember that we talked about extrinsic risk factors like UV light, smoking, obesity. Many of the cancers mutations are caused by those extrinsic factors, but we also have some factors that may also just be a part of you from birth. And so you want to make sure, in particular, if you know you have a particular mutation, that you remove those other factors that are in your control. Let's take a look at the BRCA1 risks for cancer. If we just take a look at this part right here, these individuals in yellow have the BRCA1 mutation, and in blue, they do not. What this shows us that by age of 50, your chances of developing breast cancer are 50% if you have the broken gene, as opposed to 2% when you don't. Of course, as we age, you're more likely to develop cancers, and you can see that the increased risk is up to 90% by age 70, as opposed to 77% for those individuals who do not have the BRCA1 gene. This would then indicate to us that BRCA1 testing is really important. If it's such a huge determinant of whether people will get cancer or not, you would like to know in advance of the cancer that you actually have the gene within you. Now, another maybe sort of odd thing about this is that men can also develop breast cancer. It's not as likely to happen, but the gene is on chromosome 17. Both men and women have chromosome 17. This also tells us that these other factors play a role in developing cancer. So, do you have a gene? And do you have hormones such as estrogen, which we know increases our risk of developing cancer, whereas men don't have that combination to the extent that women do. So it's less likely, but still possible that a man can develop breast cancer. And so this just shows us that maybe we can pass those genes on to our children, where we're not passing on cancer per se, but the predisposition to developing cancer. Let's now move on to the 4th hallmark of cancer, that is evasion of apoptosis. In this slide, we can see that when cells divide, they, after mitosis, now have 2 copies, and in this case, the graphic is showing you that you have 2 healthy cells. We go through another cell division, and for whatever reason, one of the cells is healthy and the other one has damage. Remember, we mentioned that there are a host of proteins in the gap which are going to evaluate this damage, and they're going to decide whether they can fix that damage. When it's figured out that they cannot, the cell should go through cell death called apoptosis. There are many genes involved in apoptosis, and what cancer does is it often ignores those because of mutations in the genes linked to apoptosis. And what will then happen is that one damaged cell will continue to grow passing on its damage and its mutations again and again and again. That's what leads to this tumor growth and metastasis. The cells also are they grow very quickly, they consume a lot of glucose, and they don't have the function that they normally should have. There's a lot of detail in apoptosis that we're going to skip over for this class. Let's move on to cells growing out of control. HeLa cells still growing decades later. We call this limitless replicative potential. And we'll give you one example of how that can happen. This is the third class of genes, proto-oncogenes. The other two were tumor suppressor genes and oncogenes. Proto-oncogenes are genes that make proteins involved in cell growth, in cell-to-cell interactions, how they communicate with each other to know whether there's too many cells around them so that they shouldn't even bother to mitose anymore. And one of the proto-oncogenes creates a protein called cyclin. And cyclin might not be mutated in and of itself, but if we make too much cyclin, we're going to push through the cell cycle and go through mitosis more often than we normally would in a non-cancerous state. This next slide tells us about something we didn't discuss under mitosis. In mitosis, we have this thing called the M phase, mitosis phase promoting factor. And that factor is composed of two different parts. One is the cyclin that we talked about, and the other one is a kinase. It's called CDK, which stands for cyclin-dependent kinase. So this kinase, which is going to start mitosis or the M phase, needs cyclin to bind to it. When cyclin is not there, the kinase is not doing its job and mitosis does not happen. Now, we need a certain balance of the amount of cyclin that's around, but as we mentioned, in certain cancerous states, if there's an overabundance of cyclin, there's an overactivity of this M phase promoting factor, and more mitosis happens, which leads to cancer. That's an example of proto-oncogenes leading to cancer. I also just wanted to mention here that there is an enzyme called telomerase. This picture down here shows us a chromosome, a graphic of a chromosome, and this end here is called a telomere. The ends of chromosomes are capped, if you will, in sequences of DNA that are called telomeres. And because of DNA replication and the lagging strand and the Okazaki fragments, we discussed before that we need this enzyme telomerase to help us keep the ends of our chromosomes long. As we age, as our cells age, there's less and less of the telomerase available, and so the telomeres become shorter over time. That's what normally is supposed to happen. However, studies have shown us that in cancer cells, telomerase is particularly active. Since it's active, it is keeping the telomeres long, and it's telling the cell, you are young, and young cells divide. So cancer cells think they're young. The telomerase is telling them at the genetic level that they're young because their telomeres are long. So that's another thing we might be able to target, is the telomerase that's keeping cancer cells young. The last hallmark of cancer that I want to talk about is sustained angiogenesis. Angiogenesis is the process by which a tumor, which is shown here in kind of this dull yellow color, convinces the cells of the blood vessel to start to divide and branch off and feed the tumor with these blood vessels, which are providing the nutrients, the oxygen, the glucose that the tumor cells need to divide. So new blood vessel growth is what angiogenesis means. I have a shark in here because we've learned that sharks very rarely develop cancer, and we've learned that they actually have a molecule that they make that prevents this branching off. It prevents angiogenesis. So even if some cells start to grow out of control in a tumor, they would be starved to death because they would not have any blood vessels feeding them. From that, we've developed a medication to help starve tumors in human patients. We'll discuss that in another lecture. That is all that we're going to talk about in regards to the hallmarks of cancer. In the next lectures, we'll talk about how we detect cancer, how we treat cancer, and we'll focus on some specific medications that are out there and how they work. Thank you very much.